So today I'm going to talk about one project that I'm currently working on. It's ongoing projects with my students. So this presentation will be a good opportunity to discuss the future directions as well. And so this is a adaptive health sessions. So I think my research data source is not directly from some sensors, some wearable devices. But I think the collected data is also multimodal time series. In terms of that, so I think these studies can also be applied to these adaptive health applications in terms of the methodology. So yeah, let's start. Today I'm going to talk about Alzheimer's disease. So people's talking about Alzheimer's disease a lot, and there's so many researches going on. Still, we have very limited knowledge in Alzheimer's disease. So Alzheimer's disease is one disease in the top 10 cause of death in US, and it's still without any treatment. This is the only disease that without the treatment in the top 10 disease. So it's neurological disorders, it's neurodegenerative disorders, the slowly some bad proteins accumulate in the brain and also show some like brain degeneration, for example, like neural injury in the brain. And also in the late stage, we are seeing a lot of like cognitive impairment in the older adults. So it's pretty much common in older adults, more than like 6 million of older adults experiencing affected by this Alzheimer's disease. So actually, let me see, I can. Yeah, from these figures, you can see in this curve, there's a multiple stages of this disease progression. It spans more than 10 years from the beginning of the, at this time point, that's the starting some bad proteins accumulated, and at the stage where we diagnose this Alzheimer's disease. So multiple biomarkers are involved, and it takes more than like decades. So really difficult to research because of these heterogeneities. Yeah, some patients might have some amyloid beta, but some patients with this bad protein, sometimes without any cognitive symptoms, or sometimes patients have some neural degeneration, but without any cognitive symptoms. Some patients have none of these biomarkers, but still have cognitive symptoms. So it's really complicated based on this combination of biomarkers. So because of this heterogeneity, developing some treatments, some disease-modifying treatment was really difficult. So I wrote some example. Maybe you heard about this medication, this very controversial medication. This is aducanumab, recently approved by FDA. It's from Yejin. This is a disease-modifying medication for Alzheimer's disease. It's known to be the first disease-modifying medication for Alzheimer's disease. So given that there's no medication in Alzheimer's disease for several decades, this is really great news to all these, all the others affected by Alzheimer's disease. But it caused a lot of controversy. Some clinics actually officially announced that they're not gonna prescribe aducanumab to the older adults. And there's many research why some discussions that we should stop using aducanumab. Yeah, so many controversy behind this aducanumab. And the reason being is like that. So there's a behind the story. I think that Dr. Charles Green described very good clinical trial. So let me talk about the clinical trial one more time here. To get approved, to certain drugs to be approved, there should be some clinical trial. There were there's a phase 3 clinical trial, 2 phase 3 clinical trial for this aducanumab. The study 301 and the study 302. So for study 302, it shows really good effectiveness. The effect size was really significant when compared to the placebo, to the treatment group. But there's one more phase 3 trial, which is study 301, but it didn't show any significant effect size. But still, Biogen gets approval for this medication. What's going on behind is that the Biogen looked into their data, and they found that certain subgroup was highly concentrated in treatment group, and that group was rapid progressor. Here. So the rapid progressor is one subtype of Alzheimer's disease. It's, like underlying biological mechanism is not very well known, but people think that these types of patients progress very fast compared to the others, so it's really difficult to treat. And Biogen think that by chance, those rapid progressors, there are more rapid progressors in the treatment groups, so that's why their effect size was not very good. So they exclude those patients and recalculate their effect size, and they claim that it has certain effectiveness. And actually, FDA approved that. So yeah, that's why there's a lot of controversy going on. They're still investigating. So the lesson learned we can learn from this example is that it's really difficult to develop medication, develop treatment without understanding these heterogeneities. So we found that the heterogeneity in these progressions, in Alzheimer's disease progressions, make us really difficult to develop treatment. So our research team believe that we can address this change, we can help addressing this challenge by organizing these heterogeneous patients into more homogeneous subtypes like this for targeted treatment. Then how can we do this? So we're going to do this by data-driven way. We're going to do this using machine learning method. So this is the motivation of this study. Of course, there's a lot of existing study that try to identify subtypes of Alzheimer's disease. I think the most established one would be NIA's classification based on these three biomarkers, based on the amyloid beta, tau, and neurodegenerations. This is well-established classifications based on the biology. But the thing is, this categorization is basically all possible combination, and it doesn't involve some specific progressions by specific features. For example, it says some neurodegeneration, but there's a multiple region in the brain, but they didn't specify certain regions. So we try to complement these knowledge-driven subtypes by data-driven analysis. And also, there's a lot of research trying to identify some data-driven subtypes. So for data-driven analysis, I think there are two components. One is data, and the other one is method. So let's look into the data first. So there's a lot of studies working on clinical registry data to identify the subtypes. And the most famous data set is ODNI and NACC's. It's a really great data set from the multi-site institutions. It advances a lot of science in Alzheimer's disease. But still, the data quality is not very good. So there are some machine learning competitions like 3 or 4 years ago using this ODNI data. The test was predicting the next cognitive score using the previous score. And it turns out that there's no machine learning method better than random guess. So the cognitive score and the data is really volatile and noisy. It's difficult to investigate some data-driven approaches. So our team, thought the other way. So we're trying to use other source of data. We try to use patient in the placebo arm in completed clinical trial. Of course, clinical trial has a limitation. It's not real-world data. It's only like maximum 2 or 4 years, not much diversity. But still, it provides very close and high-quality observation of this patient within that trial period. For method aspect, there's a lot of existing study working on these subtypes, and most study use clustering method, of course, and their focus was cross-sectional clustering method like k-means. But the thing is, our data is multimodal time series, which is it's really difficult to get the similarity between the instance. And basically, the k-means is built upon those pairwise similarity. And also, it's less reproducible, which is the most critical thing in this scientific finding. So our team tried to customize machine learning models to address this challenge. So we developed novel time series clustering method, and also to address this reproducibility, we introduced knowledge guidance as a pivot to this clustering method. So this is an overview. So basically, starting from data and the knowledge. And the data is coming from the placebo patient in clinical trial, completed clinical trial. And method is our customized method. And the final deliverable would be subtypes. And those subtypes are devised to be discriminative, the primary outcomes of interest in clinical trial. So let's get into the data first. So this is our data. This is how our data looks like. So there are many patients, mild to moderate AD patients, and they are all in the placebo groups. And they have various observations. For example, like there are some baseline conditions when they first get screened or starting the clinical trial. And over time, there's some like cognitive measure to see the effectiveness of their interventions. So there's some primary outcomes going over here. And also we have some biomarkers and brain imaging. And also we have very sparse concomitant drug information. So from this data, you can see that the inputs are multimodals. That's it's multimodal. That's why they didn't follow the same distribution. Some data are really continuous, very information rich, but some data are really sparse. For example, like concomitant drugs, those are really sparse. And also they are sometimes irregularly sampled. It's not the same interval for all observation. And it's mixture of time invariant features, and have different level of sparsity. So to address this technical challenge, we customize our model. So we develop a novel knowledge-guided time series clustering model using deep representation learning, and the model looks like this. So basically, the model is pretty much simple. So this is the input, and this is an encoder, decoder. Basically, it's a reconstruction, the sequence-to-sequence reconstructions. Because our input is a time series, we do have time invariant features. Time variant features, we concatenate the input and the sequence data over here, and all this information will fit into the encoder. And the encoder will derive some representation. And this representation is back used to decode to the original input, like this. The reason being we are doing this is that, so basically clustering is all about similarity between the pairwise sample. And to get the pairwise sample similarity, we are able to calculate, we should be able to calculate the similarity between instances. But our data is not a very simple form. So we try to abstract all this data into a certain representation, fixed size representation, so that we can calculate the similarity on top of this representation. So this is our basic format, but the thing is, this representation learning is not for clustering. It's really good for reconstructing the original input, but not for clustering. So we add a little bit of regularizations. So yeah, this is our backbone encoder-decoder using the stack-to-GLU. And we add the k-means clusterings laws on top of these reconstructions, which means that we use this representation and then use this representation to form a cluster. And this k-means clustering can be have a closed form solution using the spectral relaxations. And basically, it's a single level decompositions. We use this closed form solution as a joint optimization auxiliary task to these reconstruction models. And also, we have some knowledge on what kind of variables are important. For example, we know the primary outcomes are most important things in this data. So we try to make this representation discriminated to these clinical outcomes. So we add a little bit of supervision, and also to make this represented tight and plausible, we created some augmented data, some fake data, and let the model to discriminate this fake data. So actually do whatever we can do to make the representation better. So this is our model. So using this model, we derive cluster. Before we derive a cluster, I think I need to introduce the validation first. So we first doing the internal validation using the cross-validation, and then we are doing the external validation using the, some independent clinical trial data. So this is all about internal validation using cross-validation, so 20%, 80% splits, develop models with training, test within the test set. So we evaluate one component one another and see how this model's performance change. And we measure the performance using the clustering, how compact the cluster looks like with using the Sillier scores and UMAP plots. And also we measure that how this representation is clinically useful using the classification accuracy. It turns out that models get better as we add the components. This is the internal validation. And then we bring this model to the external data that we didn't use and follow different distribution with this training data. We set aside one trial and apply the model to the other trial. And also we test whether this clustering is not, whether this clustering is stable. So the clustering is based on the stochastic method, so there's a lot of randomness whenever we run this model, even in the same data. So we try to make sure the model is really stable. So we did a little bit of a perturbation on external data and see if the clustering is constant using AR, adjusted rand index. And if we found that it's really stable, and also the feature in each cluster is well preserved in external data. So now it's time to look into each cluster, how the cluster looks like. One bad thing about this deep representation learning is that it's black box. It's really difficult to interpret. So we develop another module to identify what kind of variables contribute the most to the clusterings. And this is the results. Maybe it can be too small, but just to highlight a little bit, there were 3 different modalities of inputs, like cognitive score, the brain volume, and amyloid beta levels. We found that the executive function for cluster 2 was the most important variable. Also, the change of the executive function was the most important, and also change of the language was the most important in the cluster 2. And we are seeing that the hippocampus volume was one of the most important regions that determine the characteristic of certain cluster 2 and cluster 1, 2, in cluster 1. And we're also seeing the amyloid beta. Amyloid beta is one of the most important variables, but in our model, we're seeing that it's equally important. So there's not so much like differences by the clusters. So maybe we can look into the variable in more details. Because of time, maybe I need to skip a little bit. But I just want to highlight a few variables. We're seeing that the language is the most important, the cognitive area that determines the progressions. And this is about the language function, how the language function different from each clusters. And we are seeing that cluster 2, the people in the cluster 2 has very well spared cognitive function in language, but the other cluster have very significant decline over the time. And this is the cluster has different characteristics in terms of their progression. And also this is a cluster characteristic in terms of this primary outcome in clinical trials. So there's 3 clusters, and we are seeing that like certain clusters have very high, for example, like subtype 1, which is the blue in this figure, and the large number means that biggest decline in this cognitive score. The CDL and ADAS-Cog is one of the most widely used primary outcome in recent clinical trial for Alzheimer's disease. And we are seeing that subtype 1 has biggest decline in this primary outcomes. And actually the subtype 2, the cluster 2, has pretty much well spared cognitive decline compared to the others. So to conclude, so it's still ongoing work, so it's really difficult to conclude and talking about some implication of this model, but we'd like to think that these novel temporal clustering models improved the traditional approaches so that we can identify certain clinically useful cluster and also reproducible with external trial data. And we also found that some of the characteristics of these clusters very well are consistent with existing knowledge, existing literatures, and also we found some new insights. So for future directions, we're trying to improve more generalizability to other clinical trial data. And now that we have this subtype, we try to estimate their treatment effect, heterogeneous treatment effect by this subtype and see are there any differences in effect size so that we can do some focused clinical trial. So yeah, that's it. Thanks so much.
Thank you very much, Dr. Kim. Any questions? Yes, many, many questions.
Hi, thank you for your presentation. You brought two concepts, and one of them was cross-validation, performing cross-validation in the clinical trial. I tried to see how we can do a cross-validation while you are actively have a different arm of a study and you're trying to recruit, and how that concept, I didn't follow that part.
I see. Yeah, that's a really great question. So, yeah, you're right. If each clinical trial have different eligibility criteria to enroll the patients, they might have different distributions. So it's unfair to compare one-to-one with different trials. So what we did is try to select a trial that shares similar eligibility criteria. So for example, like MMSE between the 20 to 26, and have the similar set of conditions that pharmaceutical company require. So we can, so before we are pulling those clinical trial data, we confirm that those data follow similar distributions. Of course, there's like differences, but relatively follow similar distribution. It's fair to compare each other's. And then we regard this pooled clinical trial as a one cohort and use that cohort to build a model, build a machine learning model, test the machine learning model, and then bring that model to the other data, other clinical trial to check the generalizability of the models.
Hello. Yeah. So I have I'm curious about two details of your model. The first question is, so you mentioned that your data is from multimodalities. Are those data paired, like the data, all different data is from the same patient, or they are unpaired from different resources?
Yeah, good question. Yeah, those data are all paired. So one specific patient have all these different modality. The frequency of record is different. Like for example, brain imaging is really not much very frequent compared to the cognitive score, but yeah, everything from the same patient.
Okay. And my second question is, so when you try to build your prediction model, how do you fusion all those heterogeneous data sets? So I know the traditional where the benchmark way is doing the RD fusion. I wasn't very sure from the illusion diagram, how did you do a RD fusion or from the input data level, or you do on the decision level?
Yeah, that's a really great question, and we actually think about that modeling choice. We're going to do the early fusion, late fusion, deep fusions, and it turns out that we use all the multimodal information at the early stages, so fuse early, and use that representation go into the GLU. Yeah.
Okay, thank you.
Let's take one more question.
So for the different progression types that you mentioned, were there different rates of sampling for each progression type of the patient, or different levels of sparsity for the different progression types that you noticed in the data set?
So you mean the different sampling rate by patient?
By patient type, as in patient type by the progression rate of each patient.
So luckily, this data is coming from pharmaceutical companies' clinical trials. So all patients are required to visit clinic at every 8 weeks. So luckily, all patients share a similar frequency in their data. And yeah, it's a little bit synthetic. It's not real world in terms of data quality. I think it has pros and cons. Yeah.
Okay, and just one last question. For the trial 2, which was generalization results that you showed, was the criteria for patient selection the same as trials 1 and 3?
Oh, that's a really good question. Slightly different. So 1 and 3 has include mild to moderate, but the external data only include moderate patients. So Discover set has a wide set of baseline condition, and external data has more focused set.
Okay, thank you.
Yeah, thank you.
Thank you very much, Dr. Kim.